Good afternoon. My name is Laura Apongo, and today my colleagues and I will be introducing our research on the genetic basis of atrazine non-monotonicity in Drosophila melanogaster. Atrazine exposure has been known to reduce the proportion of emerging adults, reduce adult survival throughout the lifetime, increase body size, accelerate developmental time, and affect male reproductive success in Drosophila melanogaster, the common fruit fly. The purpose of the study is to identify the genes that are responsible for the atrazine non-monotonicity observed within the D. melanogaster fly species population through the utilization of a genome-wide association study, also known as GWAS.
Our introduction. Atrazine is a part of a group of synthetic systemic herbicides called triazines. It is commonly used as a herbicide that causes the prevention of broadleaf weeds from growing upon crops. In general, atrazine causes negative effects on targeted plant life, such as weeds and herbs, as it disrupts the photosynthetic capabilities of these plant lives, which causes them to either dry out or to die. While there may be shown negative effects of atrazine on invertebrate species, it also can affect and negatively affect species of animals such as vertebrates in aquatic regimes or aquatic habitats. These negative effects can include, for example, demasculization and feminization of those particular species. Down below, we see atrazine exposure in our model species known as Drosophila melanogaster, which produces a range of non-monotonicity patterns.
Methods. The experiment was carried out over a 3 week period. 5 separated atrazine food concentrations were used. The female flies were separated and placed in different containers. Each of the egg-laying container was labeled with a number that corresponds to a specific fly line. For a 24-hour cycle, the model species were kept apart from their male counterparts. The female virgin flies' egg-laying response where studies use different concentration of the atrazine food with each setup. With the inclusion of the DRGB lines, the degree or non-monotonicity was assessed in use the GWAS, Genome-Wide Association Study. The result, variation in the egg-laying preference by the DGRB lines. The vertical axis on the graph represents the mean number of the egg. The horizontal axis represents the concentration of the atrazine in the food supply. As shown in the figure 1.1, we assess the DGRB for egg laying choice on different concentration of the atrazine in their food supply. The non-monotic lines is curve shape indicating the effects of atrazine concentration on the female flies, and it is the ability to lay eggs.
In conclusion, the non-monotonicity effect was very evident in the datasets since some DGRP lines expressed greater affinity to only lay their eggs in either both extremes of the atrazine concentration spectrum or in the intermediate atrazine concentrations. Some of the genes discovered to be the influencing factors for the non-monotonic dose responses in Drosophila melanogaster are CG31872, CG7368, and MSP300, and many more. Since there are many known effects on other invertebrate species due to atrazine exposure, future experimentation may be performed on the basis of discovering the genes that may influence those effects on the intended species. Or future research may also involve the effects of atrazine exposure on other physiological areas of the Drosophila melanogaster species, such as how body size may alter their relative fitness from predators in the wild. We appreciate your time in listening to this presentation, and we hope you all remain safe.